Hello everyone, my name is AJ Keefe, and in this lecture I'll be covering neuronal cholesterol metabolism and its intracellular trafficking. I'll use these concepts to then review this excellent paper titled "The GARP Complex is Involved in Intracellular Cholesterol Transport via Targeting NPC2 to Lysosomes," published in the Journal of Cell Reports. I recommend that my viewers pause the video at each new slide and read through the material and analyze the graphics before playing my commentary. And if you have any questions, please comment below and let me know. And thanks for watching. Cholesterol is an essential component of membranes, myelin sheaths, hedgehog signaling, and many other very critical cellular functions. So you may remember learning in biochemistry classes about how cholesterol helps maintain proper fluidity of membranes, and this ultimately has a very pleiotropic effect on cellular activity because proper membrane fluidity affects things like cell surface signaling, rate of endocytosis and exocytosis, motility, and just many other things. So the myelin sheaths, which envelop axons, functions to increase the speed of an action potential and the speed of its propagation, and is made up of about 20% cholesterol. And so it's also very critical for myelin sheath formation. And some signaling networks, like hedgehog signaling, require lipidation with cholesterol. And hedgehog is a protein that is critical for development, and it's called a morphogen. A morphogen is any signaling molecule or protein that affects development in a concentration-dependent manner. So the rate of diffusion of hedgehog and the subsequent concentration gradient is absolutely critical for proper embryonic development. And the reason cholesterol is so critical when talking about this is that hedgehog lipidation with cholesterol vastly improves its ability to diffuse through the extracellular matrix. The inability to produce cholesterol disrupts hedgehog signaling and causes embryonic lethality or birth defects because hedgehog can't diffuse through the extracellular matrix and function as a morphogen. In neurons, cholesterol is uniquely important for a couple of reasons. Firstly, neurons have myelin sheaths, again, that require substantial amounts of cholesterol, and those myelin sheaths need to be formed in order to propagate those action potentials. And many brain regions also depend on hedgehog signaling for developmental cues. And also, lastly, probably most importantly, neurons simply have a ton of membrane that needs to be suffused with cholesterol. And studies have shown that a lack of cholesterol in neurons greatly disturbs synaptic signaling and really just causes widespread cellular dysfunction. So the idea I'm trying to hammer home is simply that cholesterol is very important for neurons. Neurons are uniquely dependent on cholesterol. There are two main primary types of cholesterol disease and dysfunction, and they are generally grouped by either dysfunction in cholesterol metabolism or dysfunctions in transport and trafficking. And both types of cholesterol diseases result in severe neurological defects. A common disease of cholesterol biosynthesis is Smith-Lemli-Opitz syndrome, which is caused by a loss-of-function mutation in a critical cholesterol biosynthesis enzyme. A disease of cholesterol transport and trafficking that will be critical to understanding the current paper is this disease called Niemann-Pick type C. Niemann-Pick type C, or simply NPC, is caused by a mutation in the proteins NPC1 or NPC2, named after the disease. And these two proteins collectively function to transport cholesterol from endosomes to the cytosol, where they can be used. We'll discuss NPC more in the next slide, but this complex is basically required in order for cholesterol to escape endosomes after cholesterol is taken up by receptor-mediated endocytosis. And beyond these clear-cut disorders of cholesterol dysfunction, there's also a lot of evidence that suggests neurodegenerative diseases like Alzheimer's disease and Huntington's disease and even ALS are related, at least in some part, to aberrant cholesterol dynamics. In Alzheimer's disease, for example, one of the most common early onset mutations is in a protein you've probably heard of, APO, the infamous APOE4 allelic variant. You might be surprised, actually, to learn that APO is a protein that binds and transports cholesterol, and its mutation somehow causes increased risk of Alzheimer's, although I don't think it's exactly been pinpointed how, but it is associated with increased risk of Alzheimer's, a cholesterol transporting protein. You can see it in this graphic here, APO transporting cholesterol out of the astrocyte. It actually functions to lipidate itself with cholesterol. APO lipidates itself and is then transported out through the ABCA1 transporter and then diffuses. And it transports cholesterol to nearby neurons. One very important reason cholesterol dynamics are so interesting in the brain is because cholesterol cannot translocate through the blood-brain barrier. And so a neuron's supply of cholesterol is derived internally mostly from astrocytes. The rest of the body relies on the liver to produce and ship out cholesterol. But the brain can't extract cholesterol from the blood, and so it produces cholesterol in situ, or in right there at the spot in the brain. This also means that disorders of cholesterol metabolism can't rely on dietary cholesterol. And so cholesterol supplementation, just from eating more cholesterol, cannot substitute for errors in cholesterol metabolism in the brain. The neuronal helper cells called astrocytes are primarily responsible for cholesterol biogenesis, although neurons do produce a little bit themselves. But astrocytes are able to sense cholesterol levels through the ER protein called SREBP, right here, SREBP. What this protein does, it's called sterile responsive element binding protein. What it does is low cholesterol levels allow SREBP to bind SCAP, this protein, SCAP. Low cholesterol levels allows SREBP to bind SCAP, which causes trans-Golgi localization, causes it to move to the trans-Golgi from the ER. SREBP is then cleaved by resident proteases, and this releases a fragment called mSREBP, which then translocates to the nucleus and activates transcription of cholesterol biosynthesis enzymes. So during basal conditions, so just during normal conditions, when cholesterol is high and everything is happy, this protein called SCAP will bind to cholesterol and it will essentially be inhibited by cholesterol. Cholesterol's presence inhibits SCAP. When cholesterol is depleted, however, SCAP will then bind to SREBP, and this binding unmasks a Golgi localization signal, causing this SCAP-SREBP complex to translocate from the ER to the Golgi. When SCAP-SREBP arrives in the Golgi, it is then cleaved by resident proteases, S1P, S2P, and this releases the SREBP transcription factor into the cytosol, and it then translocates to the nucleus and it activates genes required for cholesterol biogenesis. And one of the main genes is HMG-CoA. I think that's what this is. And so this is how astrocytes actually sense and produce cholesterol. One of the enzymes absolutely critical for the biogenesis of cholesterol is HMG-CoA reductase. That's what the R is, sorry. HMG-CoA reductase, which controls the rate-limiting step in cholesterol metabolism. So once cholesterol is produced in the astrocyte, it gets picked up by this protein that we discussed, APO. APO lipidates itself with the cholesterol and is then secreted through the ABCA1 transporter. And now we now have cholesterol outside of the cell, outside of the astrocyte, on these APO proteins, which are then recognized by neurons. But the summary for this slide, let's just stick with the astrocytes, is that neurons are incredibly reliant on cholesterol to function properly, and they cannot get cholesterol from the blood like most cells. Instead, they rely on astrocytes to sense and produce cholesterol. And astrocytes use this SREBP, SCAP signaling program to sense and modulate cholesterol biosynthesis. In this slide, we're going to cover the canonical pathway of cholesterol import and trafficking into the neuron. So again, loss of cholesterol in astrocytes causes SREBP to translocate to the Golgi, where it gets cleaved. It then activates transcription into the nucleus, or activates translocation into the nucleus and activates transcription by binding these SRE, or sterile responsive elements, to make enzymes like HMG CoA reductase. This then increases cholesterol biogenesis. Cholesterol gets made and then it gets picked up by APO. APO then it lipidates itself and is then expelled by the ABCA transporter and is now in the extracellular matrix, ready to be recognized and picked up by neurons. So extracellular cholesterol bound to APO is recognized by the neuronal receptor LDLR, low density lipoprotein receptor, or not shown in this graphic, LRP1. LRP1 is another receptor of APO. So the binding of APO to LDLR or LRP1 causes endocytosis, receptor-mediated endocytosis. And the cholesterol ends up in these intracellular endosomes and lysosomes. They're kind of interchangeable. I'm going to use endosome and lysosome interchangeably in this. So cholesterol esters are then cleaved by various acid hydrolases. And then in order for cholesterol to become bioavailable to the cell, to the neuron, it needs to escape the endosome. And this is actually really critical. So in order for cholesterol to actually be used in the neuron, it needs to escape these endosomes. And it cannot occur by simple diffusion. It's an active process of transport. So even though the endosome is inside the neuron and the cholesterol is so close, it cannot be used until cholesterol gets out. And this protein that we discussed in the last slide, NPC1 and also NPC2, mediates the endosomal cholesterol efflux or escape. So without the NPC proteins, the cholesterol remains permanently inside of endosomes and lysosomes and is essentially locked up in these compartments, and it's not available for the neuron to be used. So not only is NPC1 and 2 critical for the endosomal escape of cholesterol, but the process by which NPC1 2 itself gets to the endosome must also be critical. So in other words, NPC1 and 2's trafficking to the endosomes is itself a critical process for cholesterol import, because if NPC1 cannot get to the endosome, cholesterol cannot get out of the endosome. Previous research has found that newly synthesized NPC1 and 2 proteins, or actually just NPC1 or NPC2, it actually requires the mannose-6-phosphate receptor for trafficking from the Golgi to the endosome. So the function of the mannose-6-phosphate receptor isn't necessarily important, but what is important in the context of cholesterol trafficking is that when the mannose-6-phosphate receptor is when this protein is not working properly, NPC1 cannot get to the endosome. So when the mannose-6-phosphate receptor translocates to the plasma membrane from the Golgi, it brings the NPC2 protein with it. And eventually NPC2 is then endocytosed and it localizes to endosomes. So the MPR, mannose-6-phosphate receptor, is critical for NPC2's localization from the Golgi network to the endosome, and it allows cholesterol efflux from the endosomes. And that now that we have a good understanding of cholesterol trafficking, we can kind of go through the paper. So NPC2 binds to MPR, the mannose-6-phosphate receptor, and is trafficked to the late endosome where it mediates cholesterol efflux. So APO-bound cholesterol enters the cell via the LDLR and the LRP1 receptors, where it requires, and it then requires NPC1 and 2 for endosomal escape. So in order for cholesterol to be successfully released into the cell, it requires both APO and NPC1 and 2 meeting each other inside of the endosome. So think of the endosome as the meeting place for both APO from the extracellular matrix that gets endocytosed and NPC1 or 2. So the question tackled in this paper is simply, what other proteins mediate cholesterol import and trafficking? In order to do this, they began with an unbiased approach that took advantage of this amphotericin screening technique that allows for positive selection of cholesterol transport deficient cells. So in other words, only cells deficient in cholesterol transport and trafficking can survive this antibiotic. And they identified 2 mutants, and following whole genome sequencing, they found that these 2 mutants contained mutations in a protein called VPS53 of the GARP complex, GARP complex. When they knocked down this VPS53 protein in wild-type cells, it prevented cholesterol escape from the late endosomes and the lysosomes. GARP complex disruption was found to be critical for the proper localization of NPC2, and this was how it was mediating cholesterol trafficking. Specifically, the GARP complex indirectly mediates proper NPC2 localization because it's required for MPR retrieval from endosomes. And remember, MPR is required for NPC2 trafficking. So the GARP complex retrieves MPR from the endosomes, and it localizes MPR back to the trans-Golgi network. And this action is required for NPC2 sorting because MPR mediates NPC2 localization to the endosomes. And then to validate these findings, which were done in culture, they then extended these findings to the whole animal. And it's really interesting because they didn't need to look very far, because an animal model of ALS, the wobbler mouse, is already known to contain a mutation in the VPS53 gene. So they took these animals and they confirmed that they show these same cholesterol deficiencies. And sure enough, in the live animal, their cells show increased cholesterol accumulation and NPC2 mislocalization. And in this graphic, we see a summary of what's discovered in this paper. We have the GARP complex down here, right here, and it's mediating the retrieval of the MPR receptors back to the trans-Golgi. We see that right here. The MPR receptors are then binding to NPC2 and moving and localizing NPC2 back to the endosome, where NPC2 mediates cholesterol efflux back into the cytoplasm. And we see that over right here. This is what happens when the GARP complex is mutated or destroyed. It causes the secretion of NPC2, because if NPC2 cannot bind to the MPR receptor for endosome localization, it's instead constitutively secreted. That's the alternative pathway of NPC2 if the GARP complex cannot retrieve the MPR protein. This amphotericin B selection method is really a slick way to select for cells that are deficient in cholesterol transport. It does this because amphotericin B binds to cholesterol at the plasma membrane and it punctures the membrane. So cells with less cholesterol at the membrane or no cholesterol will be resistant to amphotericin B. So two amphotericin B mutants were identified. They were then sequenced by whole genome sequencing, and both of these mutants had mutations that were traced back to a protein called VPS53 of the trans-Golgi localized GARP complex. shRNA mediated knockdown of VPS53 in wild-type cells caused aberrant cholesterol accumulation in the lysosomes. So we have in green we have LAMP or lysosomes, and in red we have cholesterol. When this VPS53 complex is knocked down, we have aberrant accumulation of cholesterol in lysosomes. Seeing as VPS53 knockdown causes cholesterol accumulation in endosomes and lysosomes, and this phenotype mirrors the phenotype of NPC2 mutants, they wondered if the VPS53 knockout was important, or the VPS53 protein was important for NPC2 trafficking. And indeed, they found that when VPS53 was knocked down, NPC2 was being secreted instead of being localized to lysosomes and endosomes, indicating a severe missorting of NPC2. And in general, there's a simply less NPC2 when VPS53 is knocked out. So in these Western blots, we have the two mutants. These two mutants have mutations in the VPS53 protein. And we also see there's decreased NPC2 in the whole cell lysate. But interestingly, there's a ton of NPC2 in the media around the cells. So the cells are clearly secreting NPC2 in very large quantities into the extracellular matrix. And that same phenotype is seen when they knock down the VPS53 gene or protein using shRNA. We see almost no VPS53, obviously. We see very low NPC2. We see lots of NPC2 in the media, indicating it's being secreted. We also see this is accompanied by the accumulation of, this is the NPC2 localization. So in normal control cells, we see NPC2 accumulates in these endosomes and lysosomes. When VPS53 is knocked out, we see very sparse and sporadic staining of NPC2. We also see accumulation of lysosomes. So in this slide, we're seeing more validation of the aberrant cholesterol accumulation dynamics following the knockdown of different GARP subunits. So these are all different subunits of the GARP complex. We see that cholesterol is determined by Philippin staining, is accumulating in all these different conditions. So the VPS 51, VPS 52, 53, 54, all these components of the GARP complex, when knocked down with siRNA, is causing the accumulation of cholesterol. We also see the accumulation of lysosomes, which makes sense because cholesterol accumulates in the lysosomes because it cannot escape from the lysosomes. The next question is, how does the GARP complex mediate NPC2 sorting? Well, since NPC2 sorting requires the mannose 6-phosphate receptor, NPR, they looked into NPR localization. They found that when the GARP complex was knocked down, the NPR protein was not localizing properly to the trans-Golgi network. And instead, the NPR protein was localizing to Rab7 positive vesicles, which are the lysosomes. So when VPS 53 is knocked down, we see the proportion of NPR localizing to the Golgi, indicated by P230, is decreased. There's about half as much NPR in the Golgi when VPS53 is knocked down. In contrast, when VPS53 is knocked down, we see about 20% increased amount of NPR in the lysosomes. So in other words, the NPR receptor is not making it back to the Golgi. It's stuck in the endosome, so it's not being retrieved back to the Golgi. So far, we've seen that the loss of VPS53 causes the missorting of NPC2 and its aberrant secretion, and this results in cholesterol accumulation in the lysosomes and significant cellular defects. So the GARP complex facilitates NPC2 sorting by retrieving NPR from the lysosomes, and NPR is, of course, critical for NPC2's sorting. But the question at this point is, do these studies in cell culture translate into the adult animal? Well, a previously characterized mouse model of ALS, amyotrophic lateral sclerosis, the wobbler strain, is actually caused by a mutation in the VPS53 gene, and it's already known and it's well validated. So they didn't need to look very far for a mouse model of VPS53 mutation. But the question is, does this mutation in VPS53 and the resulting pathology have anything to do with cholesterol as suggested by the current research? It turns out that wobbler mice have significantly decreased NPC2 in their cells, suggesting aberrant secretion. And indeed, wobbler mice show significantly increased cholesterol in lysosome accumulation. So in other words, a mouse model of ALS might be caused by aberrant cholesterol accumulation. And so this definitely warrants additional research. And these are the specific figures I chose to include. We have the wobbler mice right here. We see that VPS53 is downregulated because there's a mutation. Maybe it causes a splicing error. I'm not exactly sure. But we see decreased NPC2. There's significantly decreased NPC2 in the wobbler mouse strain. We also see increased density of cholesterol in cells from the wobbler mouse. We also see very significantly increased lysosomes. So it's really quite amazing that a mouse model of ALS has been traced back to a cholesterol or a mannose-6-phosphate retrieving protein that disrupts cholesterol dynamics. So the summary here is that using an unbiased screening method, the authors implicate the GARP complex in proper cholesterol transport. The loss of the GARP complex subunits, such as VPS53, causes cholesterol accumulation in the lysosomes due to NPC2 missorting. VPS53 is indirectly required for NPC2 targeting to lysosomes by retrieving NPR from lysosomes. And without VPS53, NPR is mislocalized and unable to direct NPC2 to lysosomes. These dysfunctions in cholesterol trafficking may be the mechanism by which spinal neurons degenerate in the wobbler mouse model of ALS. So the general pathway that's discovered in this paper is that GARP localizes to the Golgi. GARP retrieves NPR from the lysosomes, and NPR then directs NPC2 to the lysosomes. Pretty straightforward. And I would just like to say that I really uniquely enjoyed this paper, and I hope you guys enjoyed this video and this paper as well. If you have any questions, please comment, and I'll be sure to respond. And thanks for watching.